Okay, now the meeting is recording now. So what I was telling you now, that whenever any ion or a molecule moves in or out of the cell, it goes with certain speed, right? And what are the factors which decide the speed of the diffusion? Are these. Number one is concentration gradient. What is the meaning of gradient?
Difference.
Difference. If you have a container separating it into two halves, and here are some ions and here are some. So you can see the concentration is more here and less here. This is the difference. This difference will decide how the diffusion will take place. Diffusion will take place faster or diffusion will take place slower. So this is one factor. Factor number two is the permeability of the membrane. How permeable this membrane is. If this is a membrane, how permeable? Is it very permeable? Is it least permeable? Based on this, the speed of diffusion will be decided. If it is more permeable, definitely the diffusion will be faster. If it is less permeable, diffusion will be slower, right? Third thing, the surface area of the membrane, how big the membrane is. Suppose I cut it into like this. This is the surface area of this membrane. It has got some pores or holes, which is allowing the particles to move in and out of these two compartments. Okay. So if the area is bigger, definitely the diffusion rate will be high. Diffusion rate will be high. If the area is smaller, diffusion rate will be less. So based on the surface area of the membrane, again, the rate of diffusion will be different. Number four, molecular weight of the substance. Which substance is transported? If it is heavier, then it will move slowly, just like a very obese person. If it is crossing a road, it will be slow. If a thin person or a child is crossing, it will be faster. So the rate of diffusion will be depending upon molecular weight of the substance. So this is the fourth factor. Now fifth factor is thickness of the membrane. I told you the surface area, I told you the permeability. Now if the membrane is just like this much size or this much thickness, so movement will be easier. If the same membrane becomes thick because of edema, if there is a collection of fluid occurred in this membrane, it will take long time for the particle to move. So it is the thickness of the membrane will also decide the rate of diffusion of any substance. So these are the five factors. What are these five factors? One is concentration gradient. How much is the difference? Suppose at one place it is 10 millimoles and at other place it is 100 millimole. So there will be some diffusion. If suppose I make another scenario where the difference is here is 10 millimole and here is 20 millimoles. Now, if you see the difference here, 100 minus 10 is 90 millimoles. Okay. And here 20 minus 10 is 10 millimole. So where in this is compartment A and this is compartment B. Now you tell me in which compartment the diffusion rate will be higher. Out of
A.
A, yeah. Why?
Because they have big difference.
Big difference. Just like one is very high, one is very low. So there is a big difference. So the movement will be faster. Diffusion will be faster. Permeability, if the membrane becomes more permeable, the diffusion rate will be faster. If you increase the surface area of a substance of the membrane, it will become more easily for the molecules or ions to travel from one place to another place or diffuse from one place to another place. So concentration gradient, permeability, and area, anything increases, it will increase the net diffusion. Diffusion of the substance will be increasing. Opposite to that, the last two factors, molecular weight. If molecular weight is increasing, diffusion rate will decrease, because if a heavier substance is there, it will be taking long time to travel from one place to another place or from inside to outside the cell. Okay. Similarly, if the membrane is thin, yani thickness is less, diffusion will be high. If the membrane becomes thick, diffusion will be less. These are two opposite. Here, the two factors, they are opposite to the rate of diffusion. One increases, the diffusion rate will decrease. Here also thickness increases, diffusion rate will be decreasing. So if you combine all of them into one equation, this is an equation that rate of diffusion of substance, or it is called as Q, is equal to delta C, that is concentration gradient, then permeability, and this is the surface area, the divided by molecular weight and thickness of the membrane. So these factors 1, 2, and 3, they are directly proportional. Whenever concentration gradient increases, the diffusion rate will increase. Whenever permeability increases, diffusion rate will increase. Whenever area is increases, diffusion rate will increase. Then opposite to that, the molecular weight and thickness of the membrane, they are inversely proportional to the diffusion rate. They are inversely proportional. That means if molecular weight increases, diffusion rate will decrease. If thickness is increases, diffusion rate will decrease. Okay. Very good example of this thickness of the membrane is pulmonary edema. Pulmonary edema or pneumonia, maybe a condition where there is a inflammation of the lung is occurring. In the lung, you will have alveoli, and followed by next to it is the capillary. Here is the oxygen. Here is the hemoglobin or RBC, and here is the oxygen. So oxygen has to travel from the alveoli to the blood. This is alveoli in the lung. Alveoli. So oxygen is in the alveoli. It has to travel and transport it to the blood. If this area, this is called as the pulmonary or respiratory membrane. Respiratory membrane. If this respiratory membrane is thin, just like this, I am just drawn it is slightly bigger. If it is thin, it is easy for oxygen to travel. But if during pulmonary edema, this membrane becomes thick due to inflammation here or accumulation of the interstitial fluid. So oxygen will be slowly transported or less transportation into the blood capillaries. So that's why oxygen in the blood will be decreased. Concentration of oxygen will be decreased. So this is how it can be explained. Whenever any molecule is going in or out of the cell, two things happen. When we say sodium, what is the charge? Charge is positively charged. We say calcium, plus two charges. When we say chlorine, chloride, it is negatively charged. When we say phosphate, it is also negatively charged. So whenever any substance move in or out of the cell, it will create difference between the ions, difference between the charges, electrical charges. Just like a battery. You know the battery cell. Sorry, I am drawing it here. Sorry. Just like our battery used in the torch or somewhere, it has got positive and negative charges, right? Voltage is there, current is there, so it is positive or negative charges. Similarly, in the body, we have got sodium, chloride, phosphate, magnesium, everything is there. They are charged. They are either positive or negative. So whenever there is a movement of any ion across the cell membrane in or out, it will cause difference in the charges, difference in electrical charges. Here, it will alter the potential. It will alter the potential because inside of the cell is negatively charged and outside of the cell is positively charged. So whenever there is a movement occurring, this occurs whenever, so suppose sodium is moving out of the cell. So you are losing positive charges. You are losing positive charges. It is creating negativity inside. So we are facing a change in the or difference in the electrical potential due to diffusion. Second change, what is found whenever substance diffuses in or out of the cell, it will create a chemical change. Sorry, chemical change. The chemical molecule, sodium is moving out, or potassium is moving in, or chloride is moving out. These are chemicals. So either there will be change in the electrical charges, or could be there will be change in the chemical charges. So these are the two possibilities occur whenever any substance move in or out of the cell. It will lead to either chemical change or electrical change. What will happen? What will happen? It will create difference in the chemical nature, substances inside the cell or outside the cell, and it also create a difference in the electrical charges inside and outside the cell. This difference we call it as chemical gradient. Whenever chemical change is occurring, we say chemical gradient is created. If the positive and negative charges are changing, we say electrical gradient is created. Electrical gradient is created. But but if you see that when sodium moves out, it is chemical change also, and it is positive charge also. So it is combined, chemical plus electrical. That is called as electrochemical gradient. So whenever from a cell, any substance move out, which is electrically charged, let's say sodium moves out. This is creating chemical change. This is creating electrical change, and combined it is creating a electrochemical change or a electrochemical gradient. Why we have to remember this electrochemical gradient? Because whenever we see now in many of the places in human body, this electrochemical gradient is the cause of excitation. Thus, the tissue or the cell will get excited because of the change in the electrical or chemical gradient, like muscle. If muscle get excited, it will contract. If nerves or neurons get excited, it will send the signal. So this is because of the change in the electrical and chemical component of the cell. What is facilitated diffusion? So second point is, we have seen the passive mode of transport is diffusion. Second one is facilitated diffusion. Who will tell me the definition of diffusion? Define diffusion.
Spread out.
Yes, spreading out from.
From area of high concentration into low concentration.
Yes, area of high to area of low concentration is called as diffusion. This is simple diffusion. If it is with the help of something else, let's say with the help of a protein molecule, if it is with the help of protein molecule,
It's gonna be facilitated diffusion.
facilitate. So simply meaning of facilitation means with the help of. It is facilitated or helped by someone. So diffusion taking place, movement of any ion or a molecule from higher to lower concentration with the help of some protein is called as facilitated diffusion. So facilitated diffusion occurs due to protein channels like sodium channel, the potassium channel, chloride channel. These are the channels which will help them only for transportation. If it is high here and low here, so these channels will help them to move from higher to lower concentration. This is called as facilitated diffusion, or another name is carrier mediated transport. Carrier is something which is carrying, like a vehicle, and mediation means through. So with the help of carrier, the transportation is taking place. This is called as carrier mediated trans transport mechanism, facilitated diffusion. So the protein molecules is undergoing a conformational change here. These are the protein channel. These purple color are protein channels. Suppose this is a molecule present in the extracellular fluid, and it is in higher concentration. You can see there are many, 8 number is there, and inside is 2. So if they are separated by a semi-permeable membrane, and with the help of the ion channels, these ions will bind to this ion channel. Because of the binding, it will change its shape. Previously, shape is like this. Okay. Now, because of the binding, it will change the shape to opposite, like this. So it will open inside and closed outside. With this, the any molecule will enter into the cell. So binding of a chemical substance or ion to the protein molecule will cause a conformational change, a change which can be reversed back, and then it will move the substance into or out of the cell. This is possible both ways depending upon the concentration difference. This is called as facilitated diffusion. And so ion channels are the integral part. These channels are present in the lipid bilayer. These are the part of the lipid bilayer. And they will be traveling, they will be allowing the substances according to their concentration to move either inside or outside the cell. These channels are distinguished by two important characteristics. They are selectively permeable, means sodium channel will transport only sodium. Potassium channel will transport only potassium. So they are selective, specific for one specific chemical. And it's not only one channel will open at one time. There are so many channels, so many sodium channels can open at one time and cause movement of the ions. So these are the peculiarities of these protein channels. These protein channels also has got some gate. They are gated. So here you will see this is a sodium channel which is having gate outside. Once the gate open, the sodium will move into the cell. So this is a sodium channel, or it is causing facilitated diffusion of the sodium. Whereas potassium channel, this is a potassium channel here, which has got gate inside. Some flap like thing is there, which is opening and closing depending upon the situation. So now it is closed. Once it open, it will allow the potassium, because potassium is high inside the cell and very low outside the cell. So it will move from higher to lower side. These are the example of carrier mediated transport or facilitated diffusion. Facilitated diffusion. Facilitated diffusion.
Doctor, do all proteins have gates?
Hmm?
Do all proteins have gates?
These are protein in nature. These are protein in nature. What you are asking me? The same thing? Or?
Do all of them have gates? Like, are they closed?
No, some may be having without gate. They are just maybe like this, closing and opening. Some may have gates, like
Okay, thank you.
like a structural flap. Yeah. Okay. Coming to the third passive mode of transport is called as osmosis. Osmosis is same like diffusion, but here instead of any ion or molecule, it is the transportation mechanism for water. So if water is separated by two water solution are separated by a semi-permeable membrane, then wherever the concentration of water is high, it will move to the concentration of lower side. So if water is separated in two containers with the help of a membrane, then water will be moving from its higher concentration to its lower concentration. So same thing, which is like a diffusion. but here it is water molecule. So if you see, these are the water molecules. These are the water molecules. They are lower on this side, higher on this side. I will just go back and come again. So they are moving in the direction of from higher to lower side. Till what time they will move? Suppose we started, here is the higher concentration, here is the lower concentration, suppose. Now, if it is this membrane is permeable, semi-permeable, and water starts to move, till what time? The whole will go here or what?
Equilibrium.
Yes, till both sides will become equal. Both sides will become equal. Till that time, it will be moving. Okay? Till the equilibrium is reached. Like this. Here is these are water molecules. These are H2O molecules. Here are many. Okay? Mm. And here is pure, no, here is the water with some salt. These are not water molecules, sorry. These are not water molecules. Water is only the liquid part here. Here is pure water. Here water with salt, you can say salt. Okay. And here is pure water. Now tell me which place, A1 or 2, concentration of water is high?
2.
Yes, because here is pure water, only water. Here water plus salt. So here it is although we say it is a concentrated solution, we say here as a concentrated, and here we say diluted solution. Okay. Dilute means more water. Dilute means more water and less salt. Here zero salt. So if they are separated like this, what will happen? If the membrane is permeable, water will move from its higher concentration. Diluted means more water. The water will move from its higher concentration to the lower concentration. This is called as osmosis. Okay, suppose if you don't want the osmosis to occur, you don't want the water to move to the other side, what you will do? You will apply some pressure, some force to stop. Okay, this pressure, which the pressure which prevent osmosis, prevent osmosis is known as osmotic pressure. Osmotic pressure, because you will learn this pressure. This will come in various blocks, various places, osmotic pressure. So simply remember the definition. What is the meaning or what is the concept of osmotic pressure? Osmotic pressure is the pressure required to stop osmosis. What is osmosis? Osmosis is diffusion of water. If you want to stop diffusion of water from its higher to lower concentration, you apply some pressure, and that pressure is called as osmotic pressure. Okay. The, the force that would have to be exerted to prevent osmosis. Understood?
Yes.
So osmosis is nothing but diffusion of water through a selectively permeable membrane. So water is so small that there is so much of it in the cell and it cannot control its movement through the membrane. But it has got some channels also for water. Water also, there are some channels which are known as aquaporine channels. Aqua means water. Aquaporine channels are there. You will learn it in kidney and endocrine block. What are these and what is the importance of this? So water is moving into and out of the cell through the aquaporine channels. Now some terminologies, because we just discussed that in a solution, in a solution means some solute and water and liquid. This is a liquid and there are some solute particles. Because of this, we create a solution. Okay. This is a solution. In this solution, Based on the number of solutes, we define some terminologies. What are these terminologies? Osmoles. Osmoles is nothing but those particles which are osmotically active, the one which will decide the movement of water. They are called as the concentration of that these particles is called as osmolarity. Osmoles are those particles which will be deciding, or they are the one which are osmotically active. They help in movement of water. And osmolarity is nothing but the concentration. Osmoles in 1 liter per liter. Number of osmoles in 1 liter of water or 1 liter of liquid. This is called as osmolarity. There is another similar term with little difference. You can see R is there. Here is L is there. Osmolarity and osmolality. So only difference is osmoles, number of osmoles per instead of per liter, it will be kilogram per kilogram. This is called as osmolality. Our body has got various liquids. One of the liquid is plasma. One of the liquid is the one which is flowing in the arteries and veins is the blood, and blood contains cells as well as the liquid part which is called as plasma. So what is the plasma osmolarity? Plasma osmolarity is approximately 300 milliosmoles per liter. Milliosmoles per liter. Ranges is different. Range is between 275 to 295. But for remembrance, for understanding, for telling the things or describing, we take 300 as the 300 milliosmoles per liter as the osmolarity of the plasma. This number you have to remember, 300 milliosmoles per liter, because it will be giving you idea what is the anything above this. Becoming more than 300 will be different name, and less than 300 will be having different name. So just remember 300 is the basic level of or the normal osmolarity of the plasma. Although the range is between 275 to 295 milliosmoles per liter. Any solution, if suppose you have a solution which is having osmolarity less than 300, less than 300, we call it as less than 300, we call it as hypotonic solution. Hypotonic solution. Hypotonic solution. Here is the hypotonic solution. So, or in other words, not if you are not remembering 300, the solute concentration is less. Solution has lower concentration of solutes or diluted solution. We can say hypotonic solution. Hypotonic or diluted. Okay. Which has more water and less salt or less molecules. So if you put the RBC in it, in this solution, if you put the RBC, here RBC concentration is 300 milliosmol. And we have got hypotonic solution, the solution which is less than 300. That means more water is there. It is diluted solution, right? Diluted means more water. What will happen? Because of more water is here compared to the inside, the water will move into the cell. Water will move into the cell, and the cell will become big, huge, and may sometimes be get burst. May sometimes be get burst. So water move, sorry, I will remove this. So water will move from the solution into the cell, and cell will get burst. We call this condition as cytolysis. Cyto means cell, lysis means breakdown. Cytolysis will occur. Here is the RBC example. There it becomes bigger, swollen. What is hypertonic solution? Hyper means more concentrated. Means more than 300. You can say more than 300 milliosmol. If in this solution, if you put a cell inside this solution, what will happen? Because inside is 300 and outside may be hypertonic, may be 400. So what is happening? Here it is diluted fluid. Here it is concentrated fluid. So more water is inside the cell. So water will move out of the cell. Water is moving out of the cell. This will cause shrinking of the cell. These are the animals. These are the plant cell. But I will show you RBC. So RBC is shrinked. They will be losing water. They are losing water and they will become shrink. So result of putting a cell into a hypertonic saline or hypertonic solution, it will lead to loss of fluid from the inside of the cell, and this condition is known as plasmolysis. Plasmolysis, loss of fluid from the cell into the plasma, and cell will get shrink. Third condition is iso. Iso means same. Tonic means concentration. Isotonic means same concentration outside the cell, same concentration inside the cell. Let's say 300, 300 here and 300 here. Where will be the movement of water now?
No movement.
No movement. Very good. Excellent answer. So if both the sides same concentration is there, this is, we call it as isotonic solution. Tone is nothing but concentration. So isotonic is same concentration. So the concentration of solutes in the solution is equal to the concentration of solute inside the cell. So inside and outside of the cell becomes same. This is called as isotonic solution. And if you put the RBC in it, there will be no change in the shape of the RBC. It will remain in a state of dynamic equilibrium. Same equilibrium means both the sides will remain same. Dynamic means same does not mean stoppage of movement. There is equal amount of water is going in and equal amount of water is going out. This is called as dynamic equilibrium. Okay. Now this is an exercise. This is an exercise related to the hypertonic hypotonic solution. Now here is the solution which is having 300 milliosmol. If you put the RBC in it, what will happen?
It's isotonic.
What will happen to the RBC?
It will not
It
change.
remains the same.
It will retain its shape, normal. It will remain normal. Here it is, the osmolarity is 200 milliosmol. Is it less or more?
Less.
Less.
Less. So what is the name of this less? Hyper, hypo,
Hypotonic.
or iso?
Hypotonic.
Hypotonic. So if the RBC is kept in hypotonic solution, what will happen to the size of the cell? Water will
It
move
will
into the
swallow
cell
It
or
will
out of the
swell.
cell?
into the cell.
Into the cell, because outside it is more water, inside it is more concentrated. Concentration is 300 inside, outside is 200. So it will be moving into the cell. Cell will become swollen. And the third one, here the solution is 400 milliosmol. Is it hypo or hyper?
Hypertonic.
Hypertonic. So more concentration of water inside the cell and less outside the cell. What will happen to the water? Will move from where to where?
Outside the cell, and the cell
Yes,
will shrink.
yes, here is the answer. These are the answers written here. But in exam, you may be given one of the picture. You may be given one of the picture. and you are supposed to tell what will happen to the size of the cell. What is happening? Is it going to burst? Is it going to shrink? Or is it going to remain same? Depending upon these values. If you remember these values, 300 means iso-osmolar solution, 200 means hypotonic solution, hypo, and 400 means hypertonic solution, hyper. Okay. Okay, now identify what type of solution are these cells in. Here, which type of solution will be there? It is shrink now.
Hypotonic.
Hypo. And here, this.
Hypertonic.
Okay, in A, what is this answer? Cells are shrink now.
Hypertonic.
Hyper. Okay. In B, what is the answer?
Isotonic.
Iso.
And what is C?
Hypotonic.
Hypotonic. Here are the answers. You will see the answers down. Why not moving? Sorry. Okay. Maybe not given here, but you are right. This is hypertonic. Oh, sorry. This is hypertonic. A is hyper. Here. Hypertonic. Here it is isotonic, and here it is hypotonic solution. So in exam, you may be receiving a question based on the tonicity of the solution and what will happen to the RBC. So with this, we come to an end of the previous lecture, that is uh, movement across the cell membrane part 1, which is only passive transport mechanism, passive. What are the characteristic of passive transport? When we say passive,
It does not require energy.
very good. ATPs are not required. Then
Movement from more to less or high to
yes,
low.
like this from top to bottom, more to less. Okay, good. Examples of passive transport are, what are the examples of passive?
Diffusion.
Facilitated. Diffusion.
First is diffusion, then
Facilitated. Diffusion.
facilitated diffusion, and then last one is
Osmosis.
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 Very good. Good. Because if you don't remember, don't understand these things, in the coming block, you will not understand various mechanisms, because in the coming block, you will be not describe diffusion means what and what is the meaning of facilitated. They will just say potassium is moving by facilitated diffusion. So you have to understand it. Okay, coming to the, so those who want to score A plus, they should read from the book. Huh? And those who just want to pass, they can stick to my slides. Don't worry. No problem. If you understand, you will score A plus also. Don't worry. But you have to make a habit of reading books, because books will give you various minute points, why maybe sometimes I may not be clear in explaining. So we conclude with the lecture number 2 or part 1. I will show you now the another lecture, today's original lecture, active transport. So transport across the cell membrane part 2, it comprises of active transport mainly. Here in this lecture, what we are supposed to learn, how the energy from the ATP hydrolysis is used. Because in active transport, ATPs are utilized. So ATPs, how the hydrolysis of ATP will be used for transporting the substance against their electrochemical gradient, against. Yani although there are low concentration, but still they will move to the high concentration. Then second point is how energy from sodium potassium electrochemical gradient Because of the movement, there will be a change in the concentration, electrical change as well as the chemical change. We call it as electrochemical change will drive the net uphill the movement of other solutes. Because of these two things, sodium and potassium movement will make the movement of other solutes or other ions also. Okay. Then describe the characteristics of carrier mediated transport and distinguish between simple diffusion. We will summarize that all the two lectures. That is what is the difference between active transport, what is the difference between passive transport. And the last one is other type of active transport is vesicular transport, transportation in the form of vesicles like endocytosis or exocytosis. So these are the points which will be discussed in this lecture. So as you know, as in the previous chapter, we already discussed that passive means from higher to lower side without use of energy. But active means it has to be from low side, that is lower concentration, to the higher concentration. And it requires energy. Just like if you are on a slide here, somebody pushes you, you can come down. But if you are walking on a stair, so you have to spend energy to walk up to the stairs. It requires energy. This is ATPs are there. This is called as active transport. And this one is passive transport. So three things are there for active transport. Move the substance against the concentration gradient. Gradient means here is low and here is high. Low concentration and high concentration. Normally it is moving this way, high to low. But in active transport, it moves from lower side to higher side against the concentration gradient. Then it requires carrier molecule. It has to be through some carrier molecules. And it requires energy. It requires ATPs. Three things are there for any mechanism to be called as active transport. It is moving from lower to higher side. And you can remember this picture of a hill, and you want to go from below upward, from bottom to the top. It requires carrier molecule. Someone should take you there, and it requires energy. So for carrier molecules, we have got for the transportation to occur, it requires some pumps. What is meaning of a pump? What is a pump in Arabic? Do you know the water pump, which throws water from the below the earth to the top of the top of the building? This is a pump.
I think it's called مضخة.
Yes, I put it here. I will show you. Maybe I put it in the comments section. Discard. You will see here. Can you see? In the notes section of the slide, I put it here. In the notes section, I put some meaning, some Arabic terminology, but you know already. So protein pumps are there. So these are the channels which will be throwing any particle in against the concentration gradient. There are two types of active transport. Active transport has got two types. One is called as primary, and second one is called as secondary. Secondary active transport. Sorry. Primary active transport and secondary. Why the two divisions? When for transportation of any substance, ATPs are directly utilized. Directly. Suppose there is a molecule here. This is a channel, suppose, and this is a molecule want to go. This is a hole is the cell wall, and this is a pump. Okay. and this molecule wants to travel. If the energy is utilized here only at the pump level, this is called as primary active transport. If directly energy is utilized here only in this step, this is called as primary active transport. What is secondary active transport? Secondary active transport is energy is driven by an ion concentration gradient established by the primary active transport. So I will draw it here. Here is the pump and here is another pump. This is throwing 3 sodium out of the cell and taking 2 potassium into the cell and utilizing the energy. This is called as primary active transport. Okay. Because of throwing all the time 3 sodium is going out. The positive ions are less here now. You are only receiving 2 potassium ions. You are receiving 2 potassium ions into the cell, whereas you are throwing 3 sodium out of the cell. Because of this difference, there is another channel on the opposite side will cause movement of sodium inside the cell. Movement of sodium inside the cell because of this gradient. So if you stop this, there will be no movement of sodium on the opposite side. This is called as secondary active transport. You understand primary? Primary means whenever energy is utilized directly for transportation of any ion. Okay. This is primary. But if energy is not directly utilized there, energy is utilized somewhere else to create some concentration gradient, which will lead to easy movement of that ion. This is called as secondary active transport. When I will show you the example, you will understand. Second type of active transport is vesicular transport, which consists of endocytosis and exocytosis. We will see it later. Okay. So now we will concentrate on primary and secondary active transport. Here in active transport, movement is occurring against the concentration gradient. It is requiring energy, which is in the form of hydrolysis of ATP. When ATP molecule is there, an enzyme is there, which is called as ATPase, which will break down into ADP plus inorganic phosphate. This inorganic phosphate is nothing but the energy. This is nothing but the energy is there, which is making the movement. Okay. So molecules or ion binds to the recognition site. They have to bind to the specific site on the carrier protein, and then the protein will do the conformational change, do the change in its structure, do its change in the structure, just like a seesaw. If you put sodium here in the first step, because of the energy is utilized here, in the next step it will become like this cross, and sodium will be thrown into the cell. Sodium is coming. Suppose this is a cell, suppose. So sodium will be moved into the cell like this. Here is the picture shown. So ion to be transported is present in the intracellular fluid. Once it will bind, because you see here more ions are outside. So high concentration is outside and low inside, but still it is moving in the direction of lower to higher side. What is happening here? There is a enzyme called as ATPase, which will convert ATP into ADP and inorganic phosphate. This will cause the change, the energy is generated, and this energy will cause the binding of the ion and the conformational change. Previously, you will see the structure is like this. Now here in this direction, it is changed into the opposite way. So it is called as conformational change. This conformational change will bring the ion to the outside. This ion is gone outside. This is the ion. And again, it will come back to its original position. This is called as primary active transport because the energy is utilized on the channel directly there. Okay. Best example of primary active transport is sodium potassium ATPase channel. Sodium potassium ATPase channel, best example of primary active transport. What this channel is doing, it is present in all the cells. It is present in all the cells and it is active all the time. What it is doing, it is throwing all the time 3 sodium ions outside the cell. In replacement, it is taking only 2 potassium inside the cell. And every step there is a utilization of the energy. This is called as sodium potassium ATPase channel. Please don't forget this name throughout your life. Throughout your life, you should not forget this name. Sodium potassium ATPase pump and its function. Its function is to throw 3 sodium out and take 2 potassium in. Don't make any mistake in remembering this. Don't make. You have to remember throughout your life because this this pump will be discussed in every block, everywhere. Okay. So sodium potassium ATPase pump is a primary active transport example, great example of primary active transport. Here the energy is utilized directly at the site and it is throwing sodium outside the cell and taking 2 potassium inside the cell. So 3 sodium going out, 2 potassium coming in one cycle. And every time whenever there is ATP is converted into ADP, 3 sodium will go out because of this cell, this channel, and 2 potassium will come inside. Because of this, potassium will be higher here and sodium will be lower inside the cell. Whereas sodium will be higher outside the cell and potassium will be lower outside the cell because of this pump. It creates a steep gradient. Steep means sharp, like this, not like this. It is creating a very sharp difference, high and low. Steep gradient is created because of this, which provides energy for coupled transport of the other molecules. Because of this gradient, many other molecules will be traveled. I will show you examples and it will create electrochemical impulse. It is involved in the activity of this process, that is sodium potassium ATPase pump is responsible for electrochemical impulses. And it also promotes osmosis. It also promotes osmosis. So this is the importance here, sodium potassium pump, why it is important. It creates high osmotic pressure because of the movement of sodium, which is osmotically active particle, and protein and glucose are transported. As I told you that there is a secondary active transport. Here on this side, 3 sodium is gone, 2 potassium came in. Okay, every cycle. And on the other side, there is another channel which is called as sodium glucose channel. Sodium and glucose channel. Because sodium becomes low here, the other channel will easily transport sodium into the cell from higher to lower concentration. Once the sodium is coming, there is a coupled channel. This is glucose also. So glucose is also coming. This is how the channel is creating opportunity for other molecules to come inside. So this channel is very important. Sodium potassium ATPase channel. And whenever sodium is moving, chloride is also moving. Whenever sodium is moving, because sodium is positively charged, chloride is negatively charged. That's why they will carry together, go along one with each other. So transport of 2 or more solutes are coupled with the sodium potassium ATPase channel. Energy needed for uphill transport, that is moving from lower to higher concentration, is because of the ATPs broken down into ADP and inorganic phosphate. That's why the channel name is sodium potassium ATPase. This is an enzyme. ATP is an enzyme which is breaking down ATP to ADP and inorganic phosphate. Okay, some other terminologies. Co-transport and counter-transport. Co-transport means molecules or ions moving in the same direction as sodium. Suppose this is a cell. Sodium is moving in. Along with it, glucose is also moving in. This is called as co-transport. Both of them moving in same direction, outside to inside. Whereas in counter-transport, molecules are moving in opposite of the sodium. So if this is a cell, so sodium is moving in and maybe hydrogen is moving out. Both are moving in opposite direction. This is called as counter transport. Counter. So this is the example here shown that from the extracellular fluid, whenever we eat food, our glucose concentration into the intestinal lumen increases. When the intestinal epithelium try to absorb, it will absorb in this fashion along with sodium and glucose, and they will be moving in. Both of them are moving in this direction. So this is called as co-transport. Co-transport. Here, the channel name is SGLT1. This is intestine. The cell is intestinal epithelial cell. On the luminal side, this is a lumen. This is how. And here is the cell. So we are talking about this cell. So glucose, sodium, glucose are there when we eat. They are present in our diet. Glucose is there and salt is there, sodium is there. On the brush border of the cell of the epithelium, there is a channel which we call it as sodium glucose transporter. The S means sodium, G for GL for glucose, T for transport, and number 1. Sodium glucose transporter number 1, SGLT1. Why it is happening? It is happening because of this sodium potassium ATPase pump here. Here, sodium potassium ATPase pump removing 3 sodium out, so less sodium inside. Because of the less sodium, sodium will move from, and here is high concentration of sodium in this direction. So it will move in. If it is moving through SGLT1, it will take along with it glucose also. So this is how the glucose will be absorbed into the intestinal epithelium. Then it will leave the intestinal epithelium and reach to the blood vessel. And it will reach to the blood vessel. This is how, and both of them are moving in same direction, inside. So this is co-transport. Whereas counter transport is, this is all of them are the example of secondary active transport. No? All of them are example of secondary active transport. Previous also example is secondary active. This is also secondary active transport. Okay, counter means opposite direction. They are moving in opposite way. Because of sodium potassium ATPase pump, sodium concentration is decreasing inside the cell, right? This is throwing 3 sodium all the time and taking only 2 potassium. So sodium is less inside the cell. And if more here, high sodium concentration, sodium will try to move in, but with the help of, now the channel is different, sodium calcium channel. This channel is sodium calcium channel. This is. And the property is that whenever sodium moves in, it will throw 1 calcium out. Calcium out. So sodium calcium channel, and the direction of the movement is sodium moving in this direction, calcium is moving in this direction. So it is called as counter transport. Sorry. counter transport. Okay, both of them are moving in the opposite direction. So up till now we have seen active transport is primary active transport, sodium potassium ATPase form. Then secondary active transport we have seen co-transport and counter transport. Co means both the ions will be moving in the same direction, either inside or outside. Whereas in counter transport, one ion is moving inside, another ion is moving outside. So this is called as counter transport, and the examples we have seen. Now coming to the other active transport, which is known as exocytosis and endocytosis. This is known as a type of vesicular transport when a large amount of substance has to be taken into the cell or thrown out of the cell. We the body utilizes mechanism which are known as exocytosis or endocytosis for large molecules like hormones, okay, and maybe some nutrition. So suppose this is a cell and these are the hormone molecules are packed in a package in a vesicle. This is a vesicle. If it has to be released outside, then the mechanism is exocytosis. This vesicle will move towards the membrane, and in the next step it will bind with the membrane, and in the next step it will be detached outside the membrane like this. This is called as exocytosis. Very clear example is many hormones are released in this formation. Hormones are released by exocytosis. Okay, because we require many molecules in a bag, in a packet. Whereas endocytosis is whenever you want to engulf or take in a big molecule inside the cell, you have to utilize the process called as endocytosis, just like a bacteria. Suppose this is a bacteria. And the body tries to, or the WBC try to engulf them, take inside. So they will come near to each other. This is WBC, white blood cells. Then they will create a two arms. Just they will try to move or make a circle around it. And in the next step, they will take this inside. This is called as endocytosis. Like example is phagocytosis. Phagocytosis, where the bacteria are engulfed by the WBCs and they will be lysed. In the next step, they will be breaking down it into pieces to protect our body. So here the examples are given. Endocytosis, taking bulky material into the cell, uses energy. Cell membrane will enfold around, and then take inside. This for cell eating. This is a cell eating process. Forms food vacuoles and digests the food. This is how the white blood cells is eating the bacteria. This is the white blood cells and this is the bacteria which will be engulfed by it and then broken down into pieces. This is endocytosis. Exocytosis, see here, it is going, attaching and then releasing outside in the form of packet. Hormones are released in this form mainly. Hormones or waste materials are released in this fashion. They are present in the form of a packet. This packet will move to the cell wall and then it will open outside and it will be thrown outside. This is called as exocytosis. Again, same thing here. How it is happening. Vesicles, they will move, attached, become the part of the cell membrane and then throw the material or hormones outside the cell. Okay, in summary of the two lectures, very important table now, because it will summarize the whole process. What is Diffusion is what, osmosis is what, facilitated diffusion is what, active transport is what. So in diffusion, is it active or passive process?
Passive.
Hello, passive. So diffusion is a passive process. Molecules that move are small and hydrophobic, those which are they don't mix with water. What is the direction of movement? Direction of movement is down the
From
concentration
high to
gradient
low.
from high to low side. Very good. Do energy is required in the diffusion?
No.
No. Do we need a protein molecule? Simple diffusion. In simple diffusion, no. And same thing, facilitated diffusion. Is it active or passive process?
Passive.
Passive. Molecules are that move may be hydrophilic or hydrophobic because it is with the help of a protein or a pump. That's why no need for remembering that is it a only hydrophilic molecule or hydrophobic molecules are released. No. Again, the moment direction of the moment will be from high to low. Same like diffusion. From higher to lower side, from high to low. Do we need energy?
No.
No ATPs are required. Do we need protein pump? This is
Yes.
facilitated. Yes, it requires some help. Facilitation means help. So it requires protein pumps. Coming to the osmosis. Osmosis means, what is osmosis?
Diffusion of water.
Diffusion of water. Very good. So molecule that is moving is water. From where to where? Again, from higher concentration of water to lower concentration of water. Or you can say towards the high concentrated solution. If you have solution concentration means number of solutes will be more, water will be less. So from high to low, from diluted solution to concentrated solution. Do we require energy? No, we don't require. Do we require a protein? We don't require a protein. So this is how the passive transport. Now coming to the active transport. In active transport, this process is active process. This is an active process. Molecules that can be moved. Which type of molecule? Any type of molecule. Any type of molecule can be moved. What is the direction? High to low or what?
From low to high.
From low to high. From low to high. Or from inside to outside. Do we require energy?
Yes.
Yes, we require energy because ATPs are utilized. Do we require pump or protein?
Yes.
We require the pump because specific pump is there which is having enzyme, sodium potassium ATPase enzyme, which is causing the hydrolysis of the ATP molecule. So this is the summary taken from the book. This was our summary. This is from the book. Here you will see examples. Diffusion, for example of diffusion is diffusion of gases, oxygen, carbon dioxide, fatty acids. It is a passive process and limit of transport continues until equilibrium is reached. Until what time? When both sides will become equal. Diffusion through the protein channel. This is the facilitated diffusion, like sodium transport, potassium transport, calcium transport. These are the facilitated diffusion. It's a passive movement, but it requires a specific channel. Continues again until there is no net movement in dynamic equilibrium. Whenever both the sides will become again equal. Osmosis is movement of water. It is a passive mode of transportation. and continues until the concentration difference is abolished or until it is stopped by osmotic pressure or by opposing hydrostatic pressure. By opposing hydrostatic pressure. Carrier mediated facilitated diffusion for different types like glucose or amino acids also. For carrier mediated transport, there is always a maximum limit. There is always a maximum limit. Suppose this is a cell and these are the carrier protein for, let's say, for glucose, suppose. So although it is a facilitated diffusion and doesn't require energy, but it will be only till when all the channels are become occupied. If 3 molecules of glucose bind here, then fourth molecule will not bind because there are only 3 channels. So this is the capacity is 3 only here. Example I am giving you. So it has got some capacity that is called as T max, transport maximum, how much concentration they can transport. If it is more than that, it will not be transported. It will not be transported. There is some limit. There is some limit because of there are specific proteins required. If the number of proteins are all occupied, there will be maximum will be reached. No extra will be transported. So it has got a transport maximum. TM, transport maximum. This value is called as transport maximum. Primary active transport, we have seen examples is sodium potassium ATPase pump. It is moving the uh, ion into the opposite direction, that is against their concentration gradient. Energy it utilized directly at the level of the pump. So it is called as primary active transport. Secondary active transport, same thing as that of the primary, except the energy is not utilized at the site. Not utilized. It is utilized somewhere else. Because of their action, there is a movement occurring here. That's why it is called as secondary active transport. Also, it has got transport maximum because there are specific channels which will be transporting, like sodium hydrogen channel, sodium calcium channel, sodium glucose channel. So they have, if all of them are occupied, then there will be no more extra uh, transportation occurs. Then vesicular transport, we have seen endocytosis and exocytosis, where the bigger molecules will be taken in or thrown out like this. So this is called as endocytosis, exocytosis. Examples are release of enzymes, exocytosis, and endocytosis is engulfing the bacteria, phagocytosis. We call it as phagocytosis. Thank you very much. With this, we come to an end of our lecture number 3. Do you have any difficulty in any part?
No, thank you.
I will stop the recording now. Recording has started. How to stop? So if you did not understand anything from this lecture, please write question and send it to me. Okay, now.